Hello friends, welcome back to my channel BioFric. Let's talk about replicon and origin of replication in prokaryotes and eukaryotes. So DNA replication doesn't really start at any random location, but at a particular site, and we call it as origin of replication. Now, a unit of DNA in which an individual act of replication occurs, it's called as replicon. Okay, so each replicon has an origin at which the replication is initiated and proceeds either bidirectionally or unidirectionally. Bidirectionally is usually in bacteria. A replicon can be linear or it can be circular. So bacterial replicon, whether it may be the chromosomal replicon or plasmid replicon, it is usually circular. And in bacterial cell, the circular chromosome contains a single origin and DNA replication proceeds bidirectionally from origin. Therefore, a single bacterial chromosome, let's talk about Escherichia coli, which is about 4.6 millibase pair in size, is one replicon, that is monorepliconic. On the other hand, when we talk about the eukaryotic cells, they contain multiple replication origins on the single chromosome, and hence they have many replicons. They are multi-repliconic. That's the major difference. Now, individual replicon of eukaryotic genome are relatively small. Okay, individual replicon here, when we compare it with the prokaryotic replicon, they are smaller, and generally they are about 40 to 100 kb in size. So origin of replication is cis-acting, meaning what? So it is able to affect only that molecule of DNA on which it resides. Okay, so if this is your replicon and it will continue in this direction and both the directions, so all these elements, okay, are able to affect only that molecule. They are not transacting, they are not able to act on the other molecules. That's something called a cis-acting element, okay, sequence here. So origins are usually AT rich. Okay, why they are AT rich is because AT bonds are easier breakdown. Okay, they can be easily broken because of just two hydrogen bonds in between. So its property facilitates the unwinding or melting of the duplex DNA. So here you can see that, let's say this is your ORI somewhere. Okay, it's a replicon here. Now what happens is that these are AT rich. When they are AT rich, they can be easily separated. That means there can be easy unwinding of the DNA because less energy is required to melt this AT rich sequences than GC rich. Okay, now in E. coli, a single origin of replication present in the chromosome is referred to as OriC. Okay, in E. coli, there is just one ORI and it's called as OriC. This is the image of OriC actually. It spans approximately 245 base pairs of the DNA. That's it. Okay, so this is the structure of origin of replication in E. coli chromosome OriC. Now what it has is basically it has some repetitive base pairs. Okay, it contains repetitive 9 base pair sequences. When there are 9 base pair sequences like this, they're called as 9 mers. Here you can see 5 sets of 9 mers, and this is 13 mer. That means that there are 13 base pairs which are repeating there 3 times. So there are 13 mers. 9 mer motifs. A motif is a building site for DNA A protein and is repeated 5 times at ORC. Okay, so this is the site where your DNA A protein will go and bind. It has its own function which we'll be looking at later. So 13 mer motif repeated 3 times have the site where the melting of the DNA. Now OriC contains 2 repetitive short motifs, the same thing again, the 9 mers and 13 base pair. Okay, so a 9 mer repeat have 5 copies which are dispersed throughout OriC and binding site of a protein DNA. Now this DNA A comes along with ATP, okay, to bind there to the double helix and will melt within the tandem array of 3 AT rich 13 mer repeats. So it will bind here and it will slightly bend the DNA and then it will melt this 13 mer region. Okay, now 13 mer repeats are located at one end of OriC sequence. Okay, so that is melted. OriC also contains 11 5-GATC3-. Okay, this particular sequence is there and these are repeated 11 times. And when they are completely methylated on adenine of this GATC adenine A here, when it is methylated on adenine on both the strands, okay, it will give signal for the replication initiation. Only fully methylated origins can initiate replication. This is very important. Okay, it's kind of gene expression regulation here. Okay, only if and only if this particular double strand is fully methylated, then it will initiate the replication. Hemimethylated daughter origin cannot be used again until they have been restored to the fully methylated state. Okay, so this sequence is also important here, that is GATC. Talking about the eukaryotic ORI, the previous one, this whole ORI was of OriC E. coli, right? Now, talking about the eukaryote, let's take example of the budding yeast, that is Saccharomyces cerevisiae, also has this particular ORI, okay, origin of replication, and here it is termed as autonomously replicating sequence, ARS. Autonomously replicating sequence. A typical yeast autonomously replicating sequence is shorter than E. coli OriC. Okay, when you compare this sequence with OriC, it's shorter than that, usually less than 200 base pair. That was about 245 base pair, right? So it is less than 200 base pair, although there are 400 ARS in yeast genome, because they are repliconic, right? So they have more than 400 autonomously replicating sequences. Not all these ARS are active, okay, and ARS, although structurally diverse, they maintain the basic structure composed of 3 domains. So basically they'll have this 3 major domain: A domain, B domain, and there will be a C domain. So A domain is comprised of this consensus sequence that is called as ARS consensus sequence. Okay, this is very important. Okay, so when it comes to the eukaryote, okay, like Saccharomyces cerevisiae, it has a domain A that has a consensus sequence, okay, autonomously replicating sequence, consensus sequence. Okay, that is how you can say it. Okay, so it is ACS, and then B domain, and then we have B domain that has DNA unwinding sequences, where all those factors will come in mind and then they'll start unwinding the DNA. Okay, and C domain is important for the DNA protein interaction. What type of DNA protein interaction is occurring here? It's basically all the factors which are attaching here, all the enzymes which will come and start the replication, all these are proteins, right? And this is the double-stranded DNA. So domain C is involved in facilitating this interaction between the DNA and the proteins that will be initiating the replication. So basically A domain here, that was a consensus sequence, has 11 base pair 80 rich sequence. It's 80 rich, okay, so that it is easy to unwind the sequence. And B domain consists of 3 small accessory sequences, B1, B2, B3. Melting of the DNA occurs within B domain. However, the details of the structure of B domain differ from some ARSs. Also contain stimulatory sequence called C domain. So sometimes the C domain stimulates the unwinding, helps the B domain. A domain and B1 of B domain, okay, only this particular thing, this portion forms what is called as origin recognition sequence, that is called as ORS, okay, where bind with origin recognition complex. So there is some complex which we'll be looking at in replication part. Okay, so this particular complex comes and binds to origin replication sequence. That means it's nothing else but different types of enzymes that will unwind the DNA and start the process of replication, and they'll bind to origin recognition sequence and facilitate the initiation. So this is the ORI in eukaryotes. So each domain here has its own function. So the consensus sequence along with B1 is ORS, and B2 here acts as DNA unwinding element. So this one is mostly involved in DNA unwinding, and when it comes to domain 3, okay, is a binding site for the transcription factor, that is AB F. So ARS binding factor 1. So here when it comes to B3, this particular ARS binding factor 1 will go and bind. So that's about the two different types of ORI in eukaryotes and prokaryotes. So hope this was helpful. I have referred to the Pathfinder book.